First of all, thank you everyone for coming here. And we thank you from the Tricontinental Institute for Social Research, New Delhi office, and welcome everyone who all are here for today's talk. As you are aware that the Tricontinental Institute for Social Research is an international movement-driven institution focused on stimulating intellectual debates that serves people's aspiration. We are having our offices in 3 continents, Buenos Aires in Argentina, Sao Paulo, Brazil, that's Latin America. We are having our offices in Joburg in South Africa, and New Delhi office in India. So today we have Dr. Satyajith Rath with us, who is a doctor and one of the prominent experts in the field of biosciences. He's presently associated with Indian Institute for Science, Education and Research, which is located in Pune. And today he'll be speaking on what is COVID-19 and how do we understand it. And he would also be dealing with questions and myths related with China and how it's impacting there are young people who thinks that they won't be having any issues. So there are a lot of things which he'll be speaking about today. So Doctor, I think I'll pass over the space to you now, and we'll request you to speak. Thank you so much.
Thank you. Hello, everyone. If you have followed this series, then you've listened to Prabir Purkayastha yesterday talk about the epidemiology of COVID-19. And I'm therefore assuming that the notion of COVID-19 is now familiar for everybody. What Prabir talked about yesterday was the trajectory of an infectious disease in human communities, how it spreads, what are the patterns, how those patterns are tracked, how those patterns are analyzed, and how we understand something about the disease in terms of being able to come up with public policy. Most infectious diseases of microbial origin, in addition to this epidemiological aspect, will also have a second aspect that all of us understand as individuals more intimately related to our bodies, which is what does the microbe do to the body? How does it create disease? Where does it come from? Where does it go in terms of spreading, and how it carries out these sort of microbial molecular functions. You can call it, as Comrade Atul did in talking to me about what to speak on, you can call it the medical aspect, or if you want to be a little more technical about it, you can call it the pathophysiological aspect, meaning how the disease biology works on individual bodies. So let me speak about that a little bit, but let me speak about that in terms of trying to understand where the disease and the virus came from, how we discovered that this is a specific disease caused by one specific virus, how that discovery allowed us to generate diagnostic modalities that allowed us to track the virus and come up with the epidemiological data that we came up with yesterday in Prabir's discussion, how the virus affects the body, how we understand the basis of how the virus affects the body, what the consequences are for the body and for individual health, and of course, what is normally done about it, what can be done in the future about it, and what sort of efforts are being made. So let me start somewhat historically, that the earliest efforts, the earliest identification of this disease comes, as everybody now knows, from Wuhan in Hebei province in China. And it's a virus that was identified from a group of people suffering from viral pneumonia. And a reasonable question is, how do we identify a new virus that we've never heard of before in people who have viral pneumonia? Because we have many other viral causes of pneumonia. People are admitted with viral pneumonias to hospitals at some frequency, all the time. And you can therefore say, okay, you have patients coming into hospitals. How do you know that this is a new disease and not an old disease? So this is never certain. Essentially, what would happen is that hospitals and physicians treating hospitals and public health authorities tracking serious diseases Atul, is there a problem with audio? Because there are multiple chats saying there is no audio. So
I can hear it. Okay. Oh, people are saying they can hear it now.
Okay. All right. Thanks. So, so the question is, how do you decide when to even look for a new virus? And the answer between hospitals and treating physicians on the one hand and public health authorities on the other is, when you start seeing clusters, and a cluster is essentially more cases than you ordinarily expect to see in a short period of time, quite probably coming from places very close to each other, so geographically related, and temporally related, meaning related in time. And this is what happened from a seafood and related foods market in the city. A cluster of viral pneumonias began to arise, began to come to hospital, and eventually people began to look for what is causing this viral pneumonia. And therefore, what was done first was look for all the usual causes of viral pneumonia. This is the influenza viruses, other related viruses, other coronavirus families. And none of those showed up as being positive. As a result, what was done was one old-fashioned methodology and one newer methodology. The old-fashioned methodology was to try to grow viruses from the lung fluids of such patients. And the viruses were grown successfully on cell lines in culture, in a test tube or in petri dishes. Cells of human origin from human respiratory tissue. And viruses were identified, viruses were found by the classical methodology of growing them, of seeing them grow and damage cells, identify damaged cells, and come up with virus pictures, as well as then come up with enough virus to do the sequencing of the RNA of the virus. It turned out to be an RNA virus, and to sequence the RNA of the virus using reverse transcriptase polymerase chain reaction, or what everybody is now referring to as RT-PCR. This was done fairly rapidly, so that by early January this year, the virus sequence was recorded, and therefore everybody's begun looking at the virus sequence. So that's the identification of the virus. The obvious question is, where does the virus come from? It's a new virus. It's a virus of the coronavirus family. And the sequence and its analysis by bioinformatic methodologies in computer science allows us to look at what its closest relatives are in terms of its sequence. And its closest relatives are, as I said, the coronaviruses. Now, we are very familiar with coronaviruses. There are 4 coronavirus strains that are circulating amongst all humans. They cause upper respiratory infections, colds and coughs, and are not considered to be particularly serious infections. However, related to these 4 relatively benign coronaviruses, we have also known of somewhat more serious disease-causing coronaviruses over the past 2 decades. The first was the SARS coronavirus, the severe acute respiratory syndrome. That's what SARS stands for. The SARS coronavirus. And SARS-CoV, in the early aughts, 2002, 2003, followed a few years later by the somewhat colonially named Middle Eastern respiratory syndrome, MERS coronavirus, followed now by this third coronavirus. All 3 have interesting and significant departures in terms of what they do to the body. The benign coronaviruses infect upper respiratory tracts, and therefore you get colds and coughs, but not much else. These coronaviruses, including the coronavirus responsible for the present illness, which is now named SARS-CoV-2, SARS coronavirus 2, because it's very closely related to the original SARS coronavirus. So SARS-CoV-2 causes a disease, an illness that we are calling COVID-19, coronavirus disease of 2019. And this disease is different from the benign coronavirus diseases because the virus infects not simply the upper respiratory tract, meaning your throat, your nose, the back of your throat, and so on, but also deeper into your lower respiratory tract, meaning the branching tubes of your lungs that go deeper into lung tissue, and in fact, the lung lining cells themselves. And as a result, what you begin to get is the possibility of serious viral pneumonias where you have inflammation in the lung itself, rather than simply inflammation in the upper airways. You have inflammation in the lower airways and lungs, and as a result, you have fluid accumulation. As a result of fluid accumulation, the respiratory gas exchange that is the normal function of the lung does not happen, and as a consequence, effectively, you begin to run out of breath, and people die because they can't breathe effectively anymore. So what is the difference, and what is the difference due to? So this is related to a fairly common question that has been tossed about, particularly in social media, but in other places as well, which is, where did this virus come from? And because we have the sequence, a large number of bioinformatics groups across the world have been looking at the sequence, at related sequences, and doing extensive analysis of sequence patterns. And it turns out that this virus is related to coronaviruses that are circulating in bats. Now, bats are known to be hosts that tend to play host to a very large diversity of coronaviruses. And it's something of a biological curiosity why bat hosts have come to these apparently benign accommodations with so many coronaviruses. But there is clearly a bat coronavirus that's fairly closely related to SARS-CoV-2. What on earth is different between the bat coronavirus and SARS-CoV-2, particularly what is different that allows SARS-CoV-2 to infect people when bat coronaviruses, by and large, don't infect human cells? The answer seems to be, and from here onwards, we are all going to be referring to possibilities rather than absolutely proven facts. But the answer seems to lie in the spike protein that covers the outside of most coronaviruses. The spike protein of SARS-CoV-2 has a sequence that allows it to bind with great efficiency with a particular protein on human cells. The protein is called ACE2. It stands for angiotensin converting enzyme, but that doesn't matter. The protein is called ACE2, ACE2. And SARS-CoV-2 spike binds with quite high affinity to human ACE2. The spikes of bat coronaviruses don't. So a reasonable question becomes, where did the spike variant of SARS-CoV-2 come from? And there have been recent data over this past month or so demonstrating that pangolins or scaly anteaters, which are another mammalian host for coronaviruses, pangolins have coronaviruses, and the peculiar and unique sequence in the spike protein of SARS-CoV-2 is very closely related to that of pangolins. So a general argument has begun to emerge that SARS-CoV-2 may have originated through genetic assortment of some sort between bat coronaviruses and pangolin coronaviruses. There is a myth floating around that this is actually a human-generated virus, either generated from humans by inadvertently enhancing the virulence of the old SARS-CoV, and or by deliberately enhancing the virulence of old SARS-CoV. These are myths and they are clearly incorrect. If you simply look at sequences and analyze the sequences carefully, it's quite obvious that the way SARS-CoV-2 appears to have come about is through unintentional natural selection rather than anything particularly intentional. That's as far as the origins of the virus are concerned. How does the virus spread? The virus spreads by exchange of respiratory secretions primarily, which means through sneezes and through droplets from coughs and sneezes. A fairly rapid analysis was done 2 months ago that looked to see whether this virus can survive when sneeze droplets fall on some surface, on paper, on cardboard, on wood, steel, plaster, whatever. For how long does the virus survive? And the virus seems to survive from as short as some tens of minutes to a large number of hours, depending on what surface we're talking about. And as a result, that is a potential source of transmission of infection, which is really why we're talking about maintaining effective physical distancing by not sneezing or coughing at each other, covering our noses with appropriate masks and containment, and washing our hands so that we don't transfer virus that we've picked up inadvertently from surfaces to our own respiratory tracts. Just how much of this can be done in poor communities is, of course, a question that we all need to ask ourselves in order to generate methodologies and policies. What are the consequences of the virus infection? The consequences are that the virus enters It establishes itself in cells of the respiratory lining of the lung airways. It begins to enter and divide and make multiple copies of the virus in these cells, thereby beginning to damage the lining of these airways and of the lung. We then have the immune response of the body beginning to cause inflammation in an effort to contain and eliminate the virus. And this ongoing struggle between the virus to spread in the body and the immune response of the body to attempt to contain the virus creates the devastated landscape that the lungs of seriously ill patients show. This is not to say that everybody who's infected becomes seriously ill. Numbers are still quite uncertain and variable, but as many as 20% to 50% of infected people, meaning people in whom the virus has entered and established at least a foothold, 20 to 50% of such people may be remaining completely asymptomatic, meaning that they never show any signs of any illness whatsoever. And it is possible, again, not proven, but possible, that such people will contribute significantly to spreading of the virus, because since these are people that who will not even know that they're sick, nobody else knows they're sick, they don't know they're sick, and therefore transmission from them and spread of the virus to other people will occur sort of below the radar, if you will. A fairly large proportion of the remaining people infected will show only mild infection, mild illness lasting for a few days, primarily fever and cough. Keep in mind that this virus infects the lower airways, and therefore it's not so common to see a runny nose in this virus infection. Although interestingly, loss of the sense of smell appears to be emerging as a symptom of this particular virus infection, and it's a little bit unclear what the biological basis of that symptom is. But certainly a deep dry cough and fever for a few days are likely to be the commonest manifestations of illness caused by this virus. In about 10% odd of instances, the virus will cause lower respiratory damage to the point of causing some degree of viral pneumonia, and such people will begin to experience shortness of breath and will therefore need medical help in hospitals. And a fraction of these, perhaps as many as 30% of these, will not be able to maintain their breathing on their own, in the sense that they will be breathing, but they will not actually be getting in any oxygen, and will therefore need oxygenated breathing support through ventilators. At the worst case, if ventilators don't work, they will need support, if that is possible to get, through what are called ECMO interventions, ECMO interventions, which are essentially extracorporeal oxygenations. It's really a lung outside the body. And it's at that stage that most deaths from the virus will occur. There are interesting age distinctions. The virus clearly appears to be far more, causing far greater and more severe illness in the elderly than in the young. But please keep in mind that it's not that the virus does not infect young people. The virus clearly can infect young people. It is just that the severity of illness amongst young people is much less likely to lead to death than it is to elderly people. So while the individual risk from COVID-19 is greater for the elderly than from the young, the community risk of acquiring infection and inadvertently spreading it is just as intense for all of us, regardless of our age. So for none of us is it reasonable to say, oh, we are young and fit, and therefore we don't need to worry about SARS-CoV-2 and COVID-19. We all need to worry about SARS-CoV-2 and COVID-19 as human beings and as responsible citizens with community responsibilities. Very briefly, I suspect we will get into this in the question and answer, but very briefly, what are the treatments? What are the established treatments? What are the experimental treatments? And what are the specific preventives, particularly in terms of vaccines? So by and large, all the treatment for COVID-19 is supportive. In other words, you treat the symptoms. If you have fever, you are treated with drugs that reduce fever. If you have a cough, you're treated for the cough. If you have shortness of breath, you're treated by giving you oxygen, by giving you ventilator support, in whatever means possible. If and when people have serious pneumonias, you end up needing to have the sheer inflammation in the body to be, you have the inflammation in the body needing to be controlled, and there are a large number of methodologies that are used for that. None of those are antiviral directly. Currently, we do not have established proven antiviral drugs for SARS-CoV-2. As a matter of fact, we don't have, with the great exception of HIV, too many antiviral drugs that work anywhere near as spectacularly as antibiotics work for bacterial diseases. And there are a number of interesting biological reasons why this makes sense that we can get into if anybody's interested. But the fact of the matter is, we don't have proven antivirals. There are a large number of antivirals, particularly antivirals that were originally designed for the original SARS from 15, 20 years ago that are currently being tried. There are some HIV-related antivirals that have been tried, as far as I know, to no great success so far. There are other drugs, such as those that are called game changers by the self-styled leaders of the free world, such as chloroquine and hydroxychloroquine, that if at all help, have only anecdotal evidence of helping. And so far, at least, I have not seen any rigorous evidence that chloroquine and hydroxychloroquine make a very substantial difference to the clinical course of COVID-19. But unlike pretty much any other epidemic in history, within weeks of the epidemic and pandemic taking hold, we have a global collaborative effort, despite all its pushes and pulls and tensions, that has begun to attempt a large number of clinical trials for a number of potential antiviral drugs. Some of them are being repurposed, meaning that they are already known drugs that are currently being tested for new uses that will shortcut their introduction. Others are clearly new drugs that are being tried out. And it remains to be seen whether this unprecedented collaborative global effort will succeed or not. Finally, is there a vaccine? And the short answer to that is, right now, there isn't a vaccine. But again, we have some idea of how this virus is prevented from taking a foothold in the body by the human immune system. We have a number of old vaccine modalities that are being repurposed for designing a SARS-CoV-2 vaccine. And at least in 2 of those cases, there are initial steps towards accelerated clinical trials in humans that are being taken. So it's not unreasonable for us to expect that in another few months, by the end of this calendar year, okay, I apologize. Let me restate my sentence. I think it's reasonable for us to expect that in another few months, towards the end of this year, we will have clear indications about a vaccine. Whether we can put the community will together to actually make an affordable vaccine available at scale across the world or not will be as much a political question as a scientific and technological one. I will stop there and I will take questions.
So there are questions already coming in on the chat now, like Tanushri and someone else has also raised. You can also see that. Do you want me to read that question to you, or you directly want to answer that from the wall? I'm after reading from the wall.
Okay, I can deal with it.
Okay.
So let me start in order of appearance. There are many theories floating around regarding the low incidence of cases in India. And what view can we take on this? So here's my worry. I'm worrying that we are attempting to analyze in the absence of data. Typically, epidemiologically, one analyzes these patterns at the end of an epidemic. During an epidemic, there are far too many local incidental factors that are controlling the speed at which epidemics take off, or outbreaks, local outbreaks take off in the course of an epidemic. For us to be interpreting those at the level of nation state boundaries, as in India or in the United States or even in China, is something of a category error. So I'm not at all convinced that the number of theories floating around regarding low incidence of cases in India are based on what will turn out to be actual fact. Some of those may well have biological basis. All of them are being offered biological basis, such as that Indian immune responses are, quote, stronger, unquote, than Western immune responses. This is by no means true. There is no such evidence of a global kind that Indians or third world citizens in general have, quote, better, unquote, immune responses. They don't. There are notions floating about that BCG vaccination, especially widespread, long-standing BCG vaccination strategies, have some role to play in low incidence of COVID-19 cases. Again, these are all sort of biologically, vaguely plausible ideas, but I, at the moment, see no reason for us to be invoking them and to be taking them particularly seriously going forward. All of these are notions and ideas that we will revisit when the epidemic begins to recede and we begin to learn durable lessons from the epidemic. At the moment, I don't think that any of them are sufficiently compelling, both on the basis of their epidemiological core and on the directness and the compelling nature of their biological plausibility, for me to be enthusiastic about. The second question is, how reliable are studies that use FAJ6 as a FAJ surrogate? I'm afraid I don't know a FAJ-based study. So this one I'm going to pass on. as one that I'm ignorant of. What I do know about surrogates is, how do we know that our immune response, let us say our antibody response, is protecting us against the virus? Because we have examples. Dengue virus infections are an obvious example, where the immune response, the antibody response, in fact makes it easier for the virus to get into cells and to infect cells. So viral infection exacerbation by immune responses and antibodies is a known phenomenon. So a reasonable worry is, how do we know whether antibodies generated by SARS-CoV-2 in humans are in fact protective or exacerbating? And one way of testing that is to take the antibodies, meaning to take serum from my blood, to, in a test tube, set up a system where you take cells, you take the virus, you put the virus onto cells in the presence or absence of the antibody, and you ask, does the antibody prevent the virus from establishing infection in cells? This is classically a virus neutralization assay, a virus neutralization test. The trouble with this is that the virus is dangerous. So can we set up a virus mimic, a virus surrogate, or what virologists would call a pseudotyped virus, where the outside of the virus resembles SARS-CoV-2, but inside it doesn't resemble SARS-CoV-2. So it's an engineered virus that simply serves as a test case of pseudotyped virus neutralization assay. And a number of laboratories, virology laboratories across the world, are setting up such assays because they can be done much more safely in order to test whether antibody responses are neutralizing or not. But I'm afraid I don't know about phage surrogates. The next question is there considerable evidence that there is an increase in zoonotic diseases, or is it that now we are detecting them more? And my guess, this is a guess, I'm afraid, but my guess is that it's a little bit of both. We are certainly detecting more. In this era of being able to do DNA sequencing, being able to analyze sequences in large scales, being able to generate technologies extraordinarily rapidly compared to my youth, certainly. It is certainly true that we are detecting diseases more rapidly, and we are able to characterize them far more extensively. Is it the case that there is simply an increase in zoonotic diseases? My guess is yes, but there are two layers to that. Is it that viruses are behaving differently? I don't think so. I think that zoonotic diseases, what we call zoonotic diseases, which is really the transfer of a virus from one host species to another host species, such as a bat virus becoming a human virus, or a poultry or duck virus becoming a human virus. This is a transition that goes on in the microbial world all the time. I don't think that itself has changed. What has changed is we have put the larger biosphere of other species under more and more human pressure, and therefore there is greater and greater human interaction at high density with very many species at a much greater frequency than used to be the case. And therefore, we are simply statistically increasing the possibilities of a zoonotic transmission event occurring. And in that sense, as I said, yes, I think that we are seeing, because of a larger and larger human footprint on the ecosystem, we are seeing many more zoonotic transfer events, simply because we are increasing their likelihood, and we are detecting more and more of them. The next question is, it is widely discussed that several countries like Netherlands, England went for herd immunity approach to tackle this deadly virus. What is a reflection on this approach? The idea of herd immunity is fairly straightforward. If a virus enters a room in which there are 20 people, all of whom are susceptible, then the virus will be able to establish a foothold immediately, because as soon as it enters the room, it is going to find a susceptible person to establish a foothold in. But if the virus enters a room in which the likelihood that the first person it will meet is already immune to it, then the virus will have no access even to the susceptible people inside the room. And that way, you will break the transmission cycle of the virus. And depending on viral properties and epidemiological properties, there are modeling approaches that will predict what percentage of a community needs to be immune to the virus in order for the virus to fail to establish a transmission cycle. And that percentage is what is commonly referred to as herd immunity, protecting not simply the people who are immune, but also the other people in the community who are not immune, who are still susceptible, but whose statistical chances of getting exposed to the virus is extremely low because of all the immune people around them. And these numbers vary from anywhere between 30% to 70%. Is this a real phenomenon? Absolutely, it is a real phenomenon. Should we be going for herd immunity? The problem with that argument is, if we know what the virulence of the virus is, and all available indications are that SARS-CoV-2 is going to cause mortality somewhere between 1 and 2% of the people it infects, then if you are going to establish herd immunity, let us say, having 50% of your population exposed to the virus naturally, and if this happens extremely rapidly, then of the 50%, meaning out of half of your population, 1% of that, meaning 0.5% of your population, is going to die, and about threefold more of that population is going to land up in hospital, desperately ill. Do we have the medical, technological, health system, healthcare, socioeconomic resilience and resources to handle that large number of seriously ill people effectively and meaningfully in such a short period of time? That's really the core question for us to ask ourselves when thinking about the so-called herd immunity approach. And all available evidence about the virulence of SARS-CoV-2 suggests that simply allowing the virus to go through the community and establish herd immunity rapidly so that we can go back to life as usual is not likely to be feasible, not because herd immunity won't happen, but because the collateral damage in terms of the very large numbers of people who will be sick and whom we cannot provide the healthcare that all individuals in our societies deserve as a matter of human rights, I don't think that a purely herd immunity-based approach is appropriate. The next question is, why are children less infected? And as I said at the beginning, it's not that the virus does not infect children. The virus, as far as we can tell, infects everybody. It is that it is causing far more serious, severe illness in the elderly. And it's a perfectly good question why it is doing so. There are other diseases where children are particularly at risk of serious illness. And in this particular disease, that does not seem to be the case. There are a large number of biological theories that I've heard bruited about in the community of scientists working on this. But I don't think that there is currently any evidence to explain why this particular virus is so much more severe in its manifestations in the elderly. The only thing that we know at the moment is the epidemiological evidence that so-called comorbidities, in other words, people not simply people who are elderly, but people who are elderly and have other health problems, problems such as high blood pressure, problems of other kinds of chronic inflammatory diseases. It's having those other conditions that seems to make SARS-CoV-2 mediated illness, that seems to make COVID-19 much more severe in the elderly. In other words, if you look at those elderly people who very strictly do not have any other health-related issues that are pre-existing, then their frequency of severe illness with SARS-CoV-2 is considerably reduced. But again, we don't really have a good biological explanation set about how and why this is so. The next question is, do vitamin C pills help the immune system for the elderly and older people? Linus Pauling, both Nobel laureate and Crank, famous as both, used to talk a great deal about vitamin C. And vitamin C has been argued to do a large number of things. And as soon as something is argued to do a very large number of apparently unconnected things, one tends to be empirically skeptical about that. So do I think vitamin C is generally good for the body? Certainly. Vitamin C is an essential vitamin and we should not be deficient in it. But I have yet to see any evidence that vitamin C in and of itself has any effects on SARS-CoV-2. Let me say something about the idea that drugs can improve our immunity. Immunity against microbes is by and large tailored to each microbe. In other words, our immune system responds differently to different microbes. And therefore, a general enhancement of immunity is by and large a meaningless notion. What we really do need is, for a given disease, that particular microbe.
So, Comet, can you hear me?
Yeah. Sorry, okay, I apologize. I had a power breakdown, and as a result, my connection is now going to be extremely unstable because now I'm on a cell phone connection. So we had dealt with the vitamin C and immunity question. Can we go on from there?
Yeah. So another question is, can hydroxychloroquine be used prophylactically by high-risk individuals?
Yes. So the short answer is no. As I said, there has been some attempt at using chloroquine and hydroxychloroquine as antivirals. This was done during the SARS days, and there are at least 2 or 3 reports now so far using it for COVID-19. None of those are formal, careful clinical trials. The one that most approaches that really does not show any great deal of protection. None of this is to say that hydroxychloroquine will not be useful. It is simply to say that it does not so far on evidence appear to be a game changer. If you look at the antiviral efficacy of chloroquine and hydroxychloroquine, it's a very modest effect, quantitatively speaking. It does have an antiviral effect, but it's a very modest effect. And therefore, I think it's exceedingly unlikely that hydroxychloroquine is going to prove to be some sort of a magic bullet that will serve as excellent prophylaxis. Certainly, at this point, there isn't good evidence to say or think that. We just really need much better, much more analysis and evidence before we can say anything about it.
So yeah, so another question is, what is the mechanism by which testing is done? Why is it so difficult to manufacture and why is it expensive?
So the way testing for a virus infection in this day and age is done, and it never ceases to delight me, is what we have in the lung or throat or nose fluid is a few virus particles. Each of these virus particles has RNA inside it. And that RNA is characteristic and unique to that particular virus. So if we could detect a particular sequence of RNA, then we would have detected the virus. And therefore, we would have a diagnostic. So 30 years ago now, a biologist named Carey Mullis came up with a way of taking a very, 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 very small amount of DNA and using an enzymatic trick to amplify this DNA so that from one copy of this DNA in very short periods of time, up to an hour or so, millions of exact or reasonably exact copies could be made. And as a result, we could overcome for viral diagnostics the core problem, which is that there are very few viruses. And since each virus has only one small strand of RNA inside it, about 25, 30,000 bases, sensitivity is a problem. So you need the polymerase chain reaction. What the diagnostic test does, therefore, is take the fluid, extract all the RNA from that fluid, convert all the RNA into DNA using an enzyme called reverse transcriptase, and then use a polymerase chain reaction that is specific for SARS-CoV-2 to amplify SARS-CoV-2 sequences that may be present in that so-called complementary DNA that has been made from the RNA. So you take the RNA, you make it into complementary DNA, and then you are amplifying selectively SARS-CoV-2 sequences. In order to do this, as I said at the beginning, what you're doing is a reverse transcriptase polymerase chain reaction. And for doing that, the technological trick that is involved requires extremely careful temperature control of the reaction, and therefore it needs equipment. The newer versions, which are called real-time polymerase chain reactions, which are even better in a variety of technical ways, need even more refined control, and therefore inevitably much more sophisticated instrumentation. And it is the instrumentation that is a major blockade. There are other ways of doing the assays. There is a test called the LAMP test that attempts to sidestep some of this and generate a point of care version of a test, but its sensitivity tends to go down according to some people. There are other ways of at least converting all of this to microscale so that it can be fitted into a transportable instrumentation package, and that too some people are doing. Nonetheless, we are still talking about having to build equipment, having to create a lot of fine biochemical reagents that will go into the test, and therefore having to manufacture and quality control all of them, and making all of these available at very large scale in decentralized fashion. This is really the technical and implementation challenge that we are currently facing with the testing.
Okay, so another question is also, is there a conclusive evidence that the increase of zoonosis could be due to environmental degradation and triggered by global warming?
So let me rephrase what I said earlier. Global warming and environmental degradation are for me part and parcel of what I'm calling the increasingly prominent human footprint on the biosphere and on the global ecosystem. And as a consequence of this, the interfaces of wildlife species and humans are inevitably increasing in frequency and in density. And as a result, what I'm calling events of zoonotic transmission, where viruses go from one host species to another, are likely to be increasing in frequency. I don't think that, as I said earlier, it is that zoonotic diseases themselves are increasing in frequency. It is simply that our effects on the ecosystem are making it more and more possible to have such events, statistically speaking.
So Gargia has asked you, any comments on geographical and seasonal variations?
As I said, I'm deeply reluctant. I'm perhaps more skeptical about this than many of my friends and colleagues are. But at this point, I'm deeply reluctant to interpret the distribution data from a pandemic that is clearly emerging at different rates and with different local trajectories across the world. Is it possible? Certainly, it is possible. There is this argument that has been put forth in a preprint from a couple of researchers at MIT in the US, saying that temperate zone countries are showing a very different pattern of transmission and illness due to SARS-CoV-2 and COVID-19 than tropical countries. Different viruses are clearly differently susceptible to environmental temperatures. So can this have a plausible biological explanation? Certainly, yes. Does it have that particular explanation? I don't know. I don't even know whether the original data are eventually going to look exactly like this. So I'm at this point quite unwilling to base any of our public policy and its implementation on these sorts of really coffee table speculations, no matter how plausible they sound. I can be a little more technical about my criticism of all of these, if you will, but the bottom line still remains that I don't think that we should be basing policy and implementation discussions on that.
Okay. So US scientists have linked BCG vaccine with lower mortality rates and COVID-19 cases. How do we view this?
Right. So all right, let's get into the technicalities, which I just said I wouldn't. One set of researchers in MIT has a preprint that says they've analyzed and ambient temperatures correlate with COVID-19. Another set at the New York Institute of Technology in a similar preprint have said BCG vaccination strategies correlate very strongly with COVID-19. If you look at that preprint, what they are calculating is deaths per million population or cases per million population. They're not calculating deaths per million cases. That's something of an omission. But even more bluntly, have these two groups corrected for each other's factors in their analysis? I haven't found any indication that they have that the BCG people, in other words, have corrected for ambient temperature, and that the ambient temperature people have corrected for BCG. These are all patterns in which you can look for a large number of correlations, and you will find a fairly large proportion of those correlations looking initially valid. And are those interesting things to keep in mind? Yes, but only for going forward provisionally in understanding the disease in its biology. Should we be basing policy on this? Should we be going out and trying to vaccinate everybody with BCG? No. Should we perhaps check it out by vaccinating a few at-risk people with BCG, given how safe BCG is? Perhaps. But I don't think that any of these speculations contribute to our policy and implementation concerns beyond this.
So the other question is, why convalescent plasma transfusion, despite being simple and likely to be effective, is not recommended widely, as we don't
Right.
have time to wait for specific COVID-19 drug?
Correct. That's absolutely spot on, and as a question, and there are two clear technical scientific reasons why we can't do it quickly and why we can't do it to large scale. Remember what I said when I was talking, that the immune response, the antibodies, for example, that are generated in some virus infections, and I remind you, dengue virus is such an infection, can actually exacerbate infection rather than protect. So we really need clear testing and evidence that convalescent sera from most people who have been exposed and have recovered do provide protection. And therefore, we need what I described during my talk as the virus neutralization assay. And we need that test to be widely available. We need some empirical evidence that says, we've tested a large number of convalescent sera. Pretty much all of them provide protective neutralizing capacity, and none of them exacerbate. Once we have such evidence, then we can be confident that when we transfer convalescent serum, we are at least not going to make anything worse. Number one. Number two, convalescent serum transfer is extremely difficult to scale for implementation. You need to be extremely careful that you're not transferring any other infections that people might have, and that limits, as anybody who has ever run a blood bank at the community level knows, that creates an enormous number of checks and balances that need to be gone through before convalescent serum can be transferred, number one. Number two, we have at the moment no idea what is the amount of convalescent serum that can be transferred to provide protection. And we are going to need analysis and tests of that before we can say, okay, this convalescent serum has been tested, it has this level of potency, and therefore X many milliliters of this serum will provide protection to one individual for X many weeks, for example. We haven't reached there at all yet. I know people are doing this across the world, but we don't have the evidence in hand yet. But even if we had all of this, I don't think that we are going to be able to offer it as a prophylaxis to everybody. We just will not have that level of blood donation to be able to afford this. So it's going to emerge, it will come about, but it will need all of these issues, the technical scientific issues, to be addressed before it can be done. And whether even after that it can be done to scale to become a major player in community level prophylaxis is open to some doubt.
Okay. So another question is, since India is not following the trajectory of Europe and China so far, are we over alarming now? What is your opinion on this?
So clearly, the only way that any of us have of breaking the transmission cycle of the virus is to keep physical distances from each other. I think that the phrase social distancing is particularly unfortunate, and it should not be used for, especially in communities that are underprivileged and that depend on social solidarity. Social distancing is not a phrase that we should be using. We should be using the phrase physical distancing and social solidarity. But physical distancing is really what we know will reduce the chances of transmission and will therefore help us break transmission cycle so that we reduce the number of seriously ill people at any given point of time. That's really the modest goal that we are trying to achieve. In pursuit of that goal, any strategy in the community that takes that forward is praiseworthy. So I really think that communities should have participations in how best to achieve within their own local relevant frameworks, how best to achieve physical distancing and how best to implement physical distancing. So should we have had a national lockdown? A lockdown is the simplest way for the state to demand physical distancing. And as such, in a purely medical, technical sense, is a perfectly appropriate response, no matter what stage of transmission we are at. On the other hand, there are consequences. How long can you keep a national lockdown going? If despite the lockdown you have growing cases, then what other tactics and strategies will you put in place? Just how much of damage, not simply economic damage to the Nifty and to other stock market indices, but how much damage to the livelihoods of ordinary working class people and communities are we causing? All of these are considerations that I think should have been and still should be part of the calculus of deciding whether a national lockdown is a good idea or not.
So there is one question, which is, can you please throw some light on COVID-19 interaction with non-human animals?
So there are beginning to be reports. Sorry, before I go to the reports, keep in mind that SARS-CoV-2, remember that SARS-CoV-2 is the virus. COVID-19 is the disease, the illness. And it helps if we are careful in calling each by their proper name. So SARS-CoV-2 came to us almost certainly from non-human species, wildlife species admittedly, but non-human species. So it's a reasonable question to ask whether SARS-CoV-2 can go from us to other non-human species, particularly pets, dogs and cats. And there are growing anecdotal reports where dogs and cats have been shown to be positive for SARS-CoV-2. I haven't as yet seen any evidence that those have caused major illness in pets. I have also not seen any evidence to suggest that it has gone back from pets into people. Is all of this plausible? Yes. Is any of it happening? There is no evidence at the moment to say that it's happening. But I do know that biologists, virologists across the world are looking at how likely it is that SARS-CoV-2 will grow in other, in the lung and airway cells of other species. To get a little bit technical about this, what we have is SARS-CoV-2 binds very well to cells of our own lung air, to human lung airway cells through a cell surface protein called ACE2. So what people are looking at is how well SARS-CoV-2 binds to the ACE2 molecule, to the ACE2 protein of other species. And so far as I know, there is not yet a report of any other species ACE2 binding with very high efficacy to SARS-CoV-2. But this is early days, so we will wait and see what data emerge.
So Ravikant asked you, what is the best way to protect from COVID-19, especially in rural areas of India and of the world?
So there are, I'm sure we have access to this. There are a large number of sustainable technology alternatives to the water and soap intensive hand washing that is being recommended as the generic approach. And I would strongly suggest that something of that sort to be used. some amount of physical distancing, at least between different households, I would certainly recommend. And let me say something about a somewhat more controversial thing, namely masks. There's a fair amount of debate about whether face masks, or more correctly, whether simply covering one's nose and mouth breaks transmission or not. And a general notion that seems to be emerging is that if I'm wearing something across my face, because it's fabric and because much of the dupattas, the scarves, the bandanas that we will use as simply face coverings will not keep the small droplets on which viruses travel away, it's really not likely to prevent me from catching somebody else's transmitted virus. It might nonetheless help if I'm infected, for my infection not to go out into the community. And therefore, I am beginning to think, a little tentatively, but I am beginning to think that it might not be a bad idea at all to say, let's all cover our noses and faces with something for a while.
So this is going to be the last question. How about remdesivir? Some countries are testing it.
Yes, remdesivir is one of a fairly long series of trial drugs that many countries are testing. As yet, I have not seen results from any of these trials. Let me say something. I don't intend to be pessimistic here, but let me remind all of us about a basic difference between antibiotics for bacterial diseases and antiviral drugs for viral diseases. You see, we're all used to the idea that if we have bacterial infections, we take antibiotics and the bacteria die and the diseases get well, and antibiotics have been, over the past century, an enormous success story. Never mind that we have caused ourselves an antimicrobial resistance problem there. But nonetheless, even the antivirals that are known to be successful. So let me give you an example. For herpetic skin lesions, acyclovir is a known antiviral drug. And yet, those of our listeners who suffer from periodic herpes outbreaks will know this. If you don't start antiviral medication, acyclovir medication, immediately you notice a lesion at the very early stages of the illness, it doesn't really work very well. Does it not kill the virus? It does kill the virus. But viral growth biology is quite different from bacterial growth biology. And perhaps as a result of that, antivirals started late during the course of infection and illness tend to have a somewhat more modest effect on the illness than antibiotics do on bacterial illnesses. So I would worry about getting excessively optimistic about antivirals. Let me remind those of us who are old enough that 10 years ago, during the last great flu epidemic, we had Tamiflu. And Tamiflu worked in the sense it's an effective antiviral drug. And yet, its ability to deal with illnesses, particularly its ability to deal with illnesses when given late during an illness, is somewhat more limited than what, as I'm saying, the ability of antibiotics to deal with bacterial diseases. So the differences in bacterial and viral biology may themselves have a role to play in limiting just how useful antivirals can be. Anti-HIV antivirals work, of course they work, but HIV is not an acute infection, it's a chronic infection. And keep in mind that those antivirals, although they work, have to be taken lifelong. So we should keep much of this in mind and temper our optimistic expectation and hope for these antivirals to be effective.
Thank you so much, Dr. Satyajith.
There is a question that popped up when I relogged in, that ICMR has announced a serological surveillance among the non-contact population to see whether Indians have an antibody to COVID-19, to throw more light on the reason of less incidence of cases. And, sure. Do I think that this is very likely? Not really. It may surprise me, but at this point, I don't see any reason to be expecting this. Is this serological surveillance a good idea? Absolutely, yes. I think it's a great idea, and I think we really should be doing it. We should have been doing it even earlier, is what I would say very enthusiastically. So I will look forward to the results of these surveillances with great curiosity.
So thank you so much, Dr. Satyajith, for being here and talking to us and answering all the questions which the people have asked you. Also, I would request people who I think they also missed out on few points. The video is being recorded. It will be on YouTube. You can go back and watch it. I would also request everyone, all my friends who are here, to please subscribe to Tricontinental Institute for Social Research on YouTube, Twitter, Facebook, and Instagram. And we do come out with weekly newsletters, dossiers, and other stuff. Please go and read that. Thank you so much, everyone. Thank you so much, Dr. Satyajith. We'll catch you up.
Thank you. Thank you very much. Can I make one last point?
Yeah.
Just a quick one, because somebody asked on the stream, why bats? And it's a question that's absolutely fascinating virologists and immunologists, and we don't have a good answer. Just keep in mind that bats are flying mammals, and many of my friends are worrying about whether that has something to do in an evolutionary sense about why they harbor so many viruses without seeming to be bothered by them. So that's an open question. And on that open question, thank you very much. Thank you all. Thank you, Atul. Bye-bye.